check plagiarism?
Hmm?
Did you check the plagiarism?
I just submit. I used my own word. I hope not.
I checked. I also forgot to check, but then after submitting, I checked.
There is no submission site.
No, I like emailed her, and then I was like, if plagiarism is high, then I will resend again. But then plagiarism was not that high. All
How did you check?
online. It
Just the
was
website.
Yeah, it was 27%, and I was like, what? Then all the reference.
I
was
think it's okay because if you use technical term and it's connected to 3, I don't know, 4, 5, they think it's plagiarism.
It's because of my like reference. Once I delete the reference.
I didn't even add reference.
I did.
I just add one, one. That's just from this article.
I don't want to take any risk. I was like, I'll refer everything.
I didn't even.
Doesn't matter. There is no format for this exam.
Because
And she will give us full marking.
please, 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 please.
Did you guys read the chapter 6?
I just like
It's
read
been,
in quick succession,
it's,
and while I was sleeping, and I got a very good night's sleep yesterday.
I think we all did.
I reached 10th page and then I slept. So after that, what happens, I don't know.
I was hoping I would have an email when I wake up that the class is either cancelled or on
Right.
Zoom. I was like, so
But I tried understanding it, I did not understand much.
it's very different. After
Too much of physics.
Yeah.
Physics.
Yeah.
Now I'm seeing a lot of cells.
Like light, wavelength.
Mhm.
But then they
So
insert a
light, wavelength, everything.
I think
can understand, but
that's why she wants to read it before.
No, but then they start talking about G protein, and then all
yeah,
it back to
it
our
backs to
our
goes
field.
back to G protein.
Yeah.
Somehow I'm not able to visualize how it looks like. They've said they've talked about rhodopsin, they've talked about the two types of cells, cone and the rod cells, but I'm not able to visualize like where it is, like starting from inside out or outside in. Because they've given the incident light. The incident light has to cross blood vessels and everything, but then
Hi.
Good morning.
How's everyone doing?
Good.
Good.
How are you doing, Dr. Campbell?
I'm okay. Yeah.
And then actually they didn't like show the whole eyes, like
Right.
whole dissection of
Yeah,
the
we
eye.
should have done that.
Yes.
I don't know if I posted. Did I post your notes?
No.
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 Sorry.
Is this your new glasses? I looks
That's
new.
that's the only remaining one.
Oh, I'm gonna have to plug in my computer.
Looks like a lost.
Let me show you something.
Crack.
Not crack. It has it has
The coating.
yeah. It's due to chemical splash. I
Hmm?
think. I was working in the lab. Something splashed.
Perfection.
Which drug did you choose, by the way?
Huh?
Like we didn't discuss that drugs. What if we submitted the same drug?
Exactly.
Hmm?
But I don't think so. My favorite, like anybody, he would have chosen because it's more virus.
No, I think we all ended up choosing
Our
drugs
area.
in our comfort area.
I think I'm the only one choosing my drug.
I was saying that for the take-home exam,
Mm.
we did not discuss the drugs
Mm.
we are.
At all, yeah.
Who's
Yeah.
taking like
Mm.
what?
So what if
But
What
I
if
didn't
two of
choose
us-
my favorite area.
you chose
Your
tumor?
favorite.
I saw
I
it.
chose you.
What class are you talking about?
Take home exam.
Ah!
Like we moved from our usual tradition of discussing among ourselves
Yeah.
Uh-huh.
what drug each one is taking.
Uh-huh.
So I was
We did not do
thinking.
that.
Yeah.
We didn't do
Everyone
it this
like-
time.
Pick
So
them
what
down their
if
own.
two of us chose the same
Ah,
drug?
that's alright. That's fine.
Ma'am, was there any pattern which we have to follow, like any size, anything
No.
like that?
No,
That's good.
Mm.
just whatever it took to
Mm.
get it done.
Mhm.
Yeah, I did it in my way, like I don't know.
That's fine. It's
Mhm.
I just want to see how you can write, basically, and how you can summarize a research article.
That's the weakest point, like writing.
Ah, that's what I wanted to see. That's all.
Yeah, I can tell you that's my.
Because I
weakest
mean, honestly, the content in this course, it's interesting and everything, but I don't think like
Mm.
testing on the content
Mm.
is really the goal of these graduate courses.
Mm.
It's more to like expose you to the content and then see how you think about
Mm.
research.
Mm.
Because we're gonna actually, the rest of the class is going to be all on you guys
Mm.
from here on out. So
Mm.
we'll talk about that in a bit.
Yeah.
I'll put in the handout for today. Yeah, I mean, it's like I won't test you on what we're going through today. In other words, it's just to expose you to this new way of thinking. This is the visual transduction, which I include this because I think it's just an interesting case, and it's one that we can study you can study the G proteins at a very individual level with this
Yes.
stuff. So I just think it's an interesting way to think about it. But so I'm trying to find the handout for you. Just one second. It'll be there in a minute. Sorry I didn't do this earlier. Where is it? Oh. Oh, no, I posted the wrong ones. Hold on. Shoot. Give me a minute here, I gotta find the right one. Oh, no. It's only 6 of you now, right?
Yes.
Yes.
Yeah.
No, 6.
6, because
Yes.
Chase on.
Yes, yes.
Oh. So did he defer his, do you know what he did?
He deferred it.
Deferred it for a year? Something?
For the spring.
Spring, I was coming in spring.
Mm.
I hope so, I hope that works for him. I'm trying to find this handout. Hmm. Sorry. I don't usually do this on my iPad, so it's kind of taking me a minute to get used to this. We're almost done. Who here can vote in the United States? Only you? No one. So make sure you vote today. Okay.
Is it evident already next year?
Actually, when is the election today? Which one is it?
This is all local.
All local ones?
Yeah.
That's what I thought, yeah.
I don't even know. I sent in my thing a long time ago. I go by mail, so I don't miss it.
I usually do too, but I forgot this time.
Yeah.
Do a quick Google search of all the candidates and then.
Right. Okay, now I have to start zooming too. Oh, jeez. I'm sorry, I'm so slow today. I need a plug. I just need Zoom so I can project what I'm writing and stuff, what we're talking about, if you want to write stuff. You don't have to. So I put the notes. Did you see the notes on blackboard canvas?
Yes.
Now? You should
Yes.
see them. I'm gonna
Yes.
throw it over here. Oops, that's not right either. Oh, come on. Okay. I'm enjoying this warm weather. How about everyone else? Nice. When's it gonna get cold? Does anyone know?
Yeah, it will become cold
When?
after the last part of the week.
Oh, really?
Weekend.
It will be in 30s.
The labs are already cold.
Yeah, well, yeah. It's gonna be all winter. I really dislike the temperature regulation in that building ever since I've been here, and I've been here since 2006. So, yeah. I'm just doing this so I can share my screen. I'm not gonna record it or anything. And we don't have Chayton anymore, unfortunately.
Recording in progress. Recording stopped.
Alright, I just have to log in over here. Okay. Sorry. Just gotta do one more thing up there. I'll make myself. Bless you. Alright. Now I should be able to do this. Gonna work? Yes. Okay. Sorry about that. Okay, so I look forward to reading through your exam for this section. Basically, we were just talking about it before some of you came in. I'm just looking forward to seeing how you write about, how you summarize scientific literature, because we were talking, this class isn't really about you necessarily memorizing all the information we give you. It's to expose you to these different different scientific literature, and to also see how you think about it, how you write about it, how you present it, etc. And then yeah, so it's not necessarily for memorization, just to give you different ways to think about how drugs work in the body and different signaling transduction pathways, and again, to see how you write. That's how I run graduate classes. I don't necessarily run them to see if you know like all the context, because I know that each of you do your own area of research, so you're going to focus on that. That makes sense. So this is just to, like I said, just to expose you to different signal transduction and different mechanisms that are used for different drugs. So this chapter, I chose to keep this chapter in because I think this is a pretty interesting case with the visual transduction. If you didn't have time to read it, that's fine, but it's all about, mainly about the rhodopsin receptor, which is actually two proteins, or a protein with a ligand put together. It's an opsin protein, OPSIN, opsin protein, along with the ligand, which is 11-cis retinol, right? So this was a really interesting case because it allowed scientists who were studying this visual transduction to see actually like G protein coupled receptors at at a single cell level, basically. The caveat, though, is that these sequence of events for the rhodopsin receptor are not what we typically see with G protein coupled receptors. So I don't know if any of you got to read through or at least skim through the chapter, what did you notice that's different about the G protein coupled signaling through the rhodopsin versus through other G protein coupled receptors we talked about?
So I believe the ligand here is already attached.
Right. So the first thing is, right, so the caveat here is not typical, right, of all GPCRs, of other GPCRs, right? And the first thing is, one reason for that is the ligand is already attached. What's another reason? What else did you notice that was different? Any of you who...
It activated by photon.
Yes,
light.
right. It's activated by light. So that's different than a ligand that's soluble or, you know. Yeah. What else? Anything else?
I guess that is one reason, like since it is activated by photons, that is one reason they have like number of receptors higher than the G protein.
Why do you say that?
Because usually other ligands would either be they have this medium wherein they can float and reach to the receptors, but light is parallel. It is incident parallel. It is not it cannot reach to the target on its own. So it has to have like multiple receptors,
right,
so that one of it gets the chance to.
Right, it doesn't diffuse. Light doesn't diffuse
right,
like ligands do through like extracellular fluid to get to the receptor, right? It travels relatively in a straight line, so it has the receptor has to be in direct like line of that, yeah, right? So because light does not diffuse, so you need to increase the number of receptors so that you have a higher chance of the receptors being activated by the light, right? Yeah, that's good. What about the effector enzyme? Did you read anything about that? About the G, what is it, cyclic GMP phosphodiesterase? Right? So usually we think of after the G protein's activated, it increases the amount of the second messenger, like cyclic AMP. In this case, it would be cyclic GMP, but it's opposite here, actually. When the receptor's activated, the amount of cyclic GMP goes down, right? Because it activates that cyclic GMP phosphodiesterase that then converts the cyclic GMP to GDP or GMP. I have the photo. It's in the GMP, right?
Yes,
Yes.
GMP.
Right?
Yes.
So that's opposite of what we're used to. We're used to the second messenger being increased. So here, actually, second messenger, which is cyclic GMP, decreases after receptor activation. All right, that's good. Can anyone think of anything else? I mean, those are the main ones that I wanted to talk about. But so it's a little bit, it's almost reverse of what we saw with the other G proteins, right? The ligand's already attached. The G protein, when it's activated, that's similar, right? You have the alpha and the beta gamma subunits. The alpha subunit's what activates the effector enzyme, which is the cyclic GMP phosphodiesterase, which is it's on the last page of your notes there. You can see the figure of, not cyclic. Yeah, cyclic GMP phosphodiesterase. Yeah, sorry, I thought I was saying it wrong. It activates that phosphodiesterase to decrease the amount of cyclic GMP. So those are the main ones. Oh, and then of course, like Vivek said, the number of receptors, it's higher than the G protein, and usually it's the opposite, right? Because we have more G protein in order to interact with more receptors. In this case, we need more receptors because of the light activates it, and light doesn't diffuse like the other ligands. Okay, good. So the ligand that's attached to rhodopsin is 11-cis retinol. It's a chromophore, which means it's just a atom or a group that is responsible for the color of a compound. So it's not a big deal. It is the ligand for rhodopsin, but it's already bound to the rhodopsin.
Man,
Yes.
I was just thinking about it. Like
Yes.
How is this like the body always creates the thing which is required for that position.
For that, for what?
Like body creates things which are required at certain things like areas.
Okay.
And same goes for the receptors as well.
Yes.
This type of receptor is required over here, so that's why this receptor is there.
Yes.
If we see over here the G protein receptor, it's not working in its usual way, and it's also ligand gated as well, but it's also not working in its usual way.
Right.
So why this receptor is over here rather than being other receptor which does not require any ligand, which works in opposite manner than to G protein?
Okay, so why are you asking why do you think that's this G protein coupled receptor?
Yeah, it was just it came logically to me, like everything is very very organized in the body, and
Yes.
what is required is there.
Yeah.
But do you think that the G protein receptor is really essential over here at this site instead of any other receptor? Even if it's ligand gated, why it's ligand gated if the ligand is already bound to it?
Right.
And why it's going in opposite direction, like there is a major difference between like the cyclic MP is not increasing with the activation of the receptor,
Right.
as it's increasing in the other areas, other
Yeah, I mean, from what I know, which is very very basic about the visual system, and pretty much what I read here and what I've learned over the years. I would say that it's because because of the fact that the visual system requires light, or like we don't see as well in dim light, right? So I think there has to be a level at these G protein coupled receptors in order to discern bright light versus medium light versus low light. So in that way, the mechanism would have to be such that it's like primed and ready to go, depending on, like, because the light changes very quickly, right? It can change very quickly. So it almost seems like the G protein coupled receptor, instead of waiting for a ligand or waiting for something to activate it, has to be kind of primed and ready to respond to whatever environment we're in. That's how I kind of think about it.
So what I get it, like the quickness of the G protein receptor makes the best receptor to be present in the retina.
Mm-hmm.
Like if we look at the speed of the working of the receptors,
Yes.
there are other receptors, like I can see tyrosine kinase receptor, which
Right.
works way more faster
Sure.
than the G protein coupled receptor.
Sure.
So if the speed is the reason, then there are other receptors which has more speed.
Yeah, but I think yes, speed, but so at least when I think of this in neuroscience and when I'm talking to the PharmD students, when you have a reaction that's very quick, like an ion channel opens, it's not going to be as discerning as a G protein coupled receptor, right? So an ion channel opening is an ion channel opening. G protein, you can fine tune the signaling, right? Because you can have different G proteins, you have different enzymes activated. So I'm thinking that it wouldn't be discerning enough for our visual system to just have ion channels or just have kinase receptors, right? It has to be something that's going to be able to make a specific signal that's going to be discern the information.
That seems more like I got that, like
maybe
we need more control as well.
Yes.
Not just the speed.
Exactly.
Maybe, and then this receptor
speed
is the
and
best.
control. Yes.
Yeah,
right.
tyrosine kinase receptor you cannot get control in it.
Right.
Ion channel you cannot get
Right, because
control in it.
tyrosine kinase is going to be. There's so many kinases. It's not it's not as specific for cell to cell, right? Ion channels open. That ion's going to come in. Doesn't matter what cell you're talking about, right? And it's going to do many things. So, yeah.
So the G protein basically is
it like specificity?
Even if it's going out of the box over here, like
Yes.
it's not working in its main, like in what it works like.
Right.
Even then, this is the most suitable receptor
Yes.
for the eye.
That's what I think. I mean, I don't know.
That's more logical, like
Yeah,
I'm
right,
just
right.
thinking
Yeah,
like,
no, it's a good-
if
yeah.
It's going so out of its way
Mm-hmm.
for working over here.
Right.
Like, then why not the other receptor is here?
Right.
that's-
And I
that
think
was-
it's just because of the information and the sophistication of our visual system has to be able to discern different information. So that's what I would think. Anyone else have any ideas?
I believe that the beauty is G protein coupled receptors, they're able to modulate the intensity of light, how much light we should perceive, how much not. Like in bright, in dark, you're still able to, of course, a few seconds you won't be able to see, but after some time you try to, at least you can locate things in like in dark.
That's the
objects in
quickness.
dark.
That's the quickness.
That's the beauty of G protein coupled receptors. I guess they also, along with speed, they also have the regulatory arm.
Yes.
Which kind of modulate the
Right.
light.
The control.
Yep. Okay. Yeah. Good thinking. I've never even really thought about that, but it's good to think like that. Okay. Oops. Okay. All right. So what happens with the 11 cis retinol is it absorbs the light that diffuses into the retina, and it's interesting if you go. But let's go. Oops. If we go back here, right? So. Just let's look at this for a second. So here's, there's the eye. We don't have, and then the optic nerve going to the brain on the top left there, and then the light that comes through. It's interesting to me too that the cells that the light needs to get to are actually further back in the retina than the nerve cells, right? So here in the back are the cones and rods. The rhodopsin is on the rod cells. The cones have a different type of opsins. They have 3 different opsins that discern between green, red, and blue. So when I'm talking about rhodopsin here, I'm talking about the rod cells, because the rod cells are the ones that allow us to see in dim and bright light. Like they're the ones that are going to respond to the light more so than, I mean, the cone cells still do, but it's more so the rods that are responding to the light. So the rod cells here then are, if you look at this next figure here, this one, right? You can see, so the cones are the ones that are blue, red, and green, because that's what they're there for, to discern the colors. And then the rods are just the gray ones here. They have this outer segment, which is actually where, let me see if I have a better picture of that. Here, So they have this outer segment here. These are the outer segments. This is the cone and this is the rod. And that's the outer segment down here, especially, is where we're talking about. There's these discs that actually they form originally as what am I looking for? Like folds into the membrane, and then they eventually pinch off and become specific discs inside the outer segment. And each of those discs has the rhodopsin molecule on them, lots and lots of. So over here it shows you here, each of these discs have that receptor on it, the 7 transmembrane G protein. So this is the opsin part of it, and then here is the 11 cis retinol. Right. Okay. So so yeah, these are like flipped. This one this one has the outer segment up here, up top, and this one has it at the bottom, where this is more anatomically correct, right? These are the epithelial cells, the pigment epithelial cells, and they are very close proximity to the outer segment of the rods and the cones. So the light coming in from the eye has to travel all the way back here in order for information to be gathered from the rod cells, right? And then the outer segment contains those discs, and then the inner segments have, what we normally see with cells, the nucleus, the mitochondria, and stuff. And then they have this synaptic connection here in the inner segment, that then, if we go back to that figure we were looking at here, that then connects to these neurons here, which are sensory neurons. They're bipolar sensory neurons. So that just means the cell body is here, and then they have dendrites coming off here and axon here, right? So it's a bipolar type neuron. Normally when we think of neurons, think of like the dendrites coming right off the cell body, but these have 2 branches coming off of them. And one is where the axon would be, and one is where the dendrites would be. So these are sensory neurons in the eye here, in this part, in the retina, and then they take information. The sensory neurons then take information to the optic. These other neurons that eventually take the information from the optic nerve to the thalamus to the visual cortex, right? So everything from our visual system is actually going into the brain through the optic nerve to the thalamus, which is the relay center for the brain, and then to the visual cortex, where we're actually making sense of the vision, what we're seeing, right? And the visual cortex is way back in the back of the head. Okay, so these are just little neurons. Okay, so that's what it looks like. So once it absorbs the light, it actually it then converts. It's converted to 11 trans retinol, and then this actually diffuses into the pigment epithelial cells. which then recycles the 11 cis retinol. So recycle from 11 trans retinol recycles through different reactions, converts it back to 11 cis retinol. So this is how they're recycling 11. So the 11 cis retinol is the chromophore, or the group that gives the rhodopsin color, and then the opsin is a colorless protein, and that's the 7 transmembrane spanning region of the rhodopsin. Okay. So that's similar to other G proteins. All right. So then the photoreceptor cells that I just mentioned are rods and cones. This was amazing to me, to learn when I was first, researching this for class when I taught it a couple years ago. In the retina, we have 120 million rods. So I just have to write this down because it's just 120 million rod cells and 6 million cones. Amazing. They have similar phototransduction, so they have like the opsin, like I said, but in the cones it's different opsins because it's for color rather than light levels. So the cones, what they are important for is what we call phototopic vision. And that just means the ability to discern colors, color discrimination. Okay. And there are 3 types, red, blue, and green. They all have 11 cis retinol embedded in them, so that's similar to what we see in the rod cells. And they actually take the absorbance of the light and convert it to either red, green, or blue. So that's how they work. And we're not going to talk too much more about those. We're going to focus on the rods. So this is the rods are specifically for sensing different light levels, right? Especially low light levels. So and this is called scotopic vision. So photopic vision in the cones, scotopic vision in the rods. In dim light, the cone cells aren't working as well, so we don't see color as well in dim light. It's mostly the rods being activated in that case. So we're going to focus on the rods. They are basically, what it says here, highly differentiated epithelial cells in which the light sensing region is segregated from the inner segment, which is the main cell body, what we think of, like I said, the nucleus, the mitochondria, all the organelles. And then the light sensing region is the outer segment. So this is another, I thought, amazing thing. So we already talked about these discs. In that outer segment, each rod cell has 1,000 to 2,000 of these intracellular discs. Amazing. And these discs have a finite life cycle. They only last a few weeks. So the discs, when they get to the distal end of the rod, they will actually eventually fuse with the membrane and just be gone. And I think if they leave any debris, then that's picked up by the lysosomes in the pigment epithelial cells. Let's see, they are only 16 nanometers thick, so they're very, very, thin. So I already said that the discs have the rhodopsin, so this is where the rhodopsin is located. And I already said that it's formed by the invagination of the plasma membrane. So it, like I said, you have a valley or a trough in the membrane, and it eventually pinches off and becomes a disc. So if this was the rod cell here, the discs here. So all around the discs, this is the cytoplasm, right, of the rod cell. Like I showed you in that figure, the discs have all those molecules of rhodopsin, and that's what's mainly made of. The discs mainly, it's like 50% of the weight of the discs are the protein rhodopsin. And a single rod cell has about 10 to the 8 molecules of rhodopsin. So what's that? That's a billion? Is that a billion?
More than
0.1 billion, 100 million.
100 million. 100 million. So each rod cell has 100 million molecules of rhodopsin. 10 to the 8, so it'd be 1, 2. 1, 2, 3. Yep, 100 million. Yep. Right?
Yes.
Or wait, maybe. Wait, I can't even see where I put my commas.
Yes.
100 million, right? Yeah.
Yes.
1, 2, 3, 4, 5, 6, 7, 8. 1, 2, 3, comma. 1, 2, 3. Yeah, 100 million. 100 million rhodopsin molecules per rod cell. So it's pretty amazing. So something that is similar, it's very similar to the other G protein coupled receptors in that it has 7 transmembrane spanning domains, and that one, the N terminal is in the extracellular space, and the C terminal goes into the lumen of the discs. That's the main thing. So I'm just trying to look at my notes here. So the G protein that is coupled to rhodopsin is called transducin. So, and it's written like GT. So this is the G protein coupled to rhodopsin. And like we said, there's far fewer molecules of the GT, the transducin, than there is the G protein. And then, like Vivek brought up, that's not what we're used to in other G proteins. And it's because of the photons of light travel basically in a straight line and cannot diffuse to other molecules. So they need to be captured by these rhodopsin molecules. The way the 11 cis, so we already said the chromophore is the 11 cis retinol. And it is attached to the opsin to a lysine in the center of the loop that is extracellular, or extra, not really cellular, extra discal outside the disc. So if we look here, hold on, I think there's a picture here. Yeah, here. So right, so this is the disc membrane here, right? Here's the opsin, which is the 7 transmembrane spanning 7 transmembrane spanning domains in it. In the last domain, which has the N-terminal loop into the cytosol of the rod cell, here's where the 11-cis retinol is, and it's bound to that loop via a covalent reaction. And we don't have to get into the chemistry of it, mainly because I can't get too far into the chemistry of it, just knowing that this is a... Sorry, just a second. It does this. I don't know why. Tried to fix it. So basically, it's like this. This is the reaction. So here is the A is the 11 cis retinol, and here's the lysine, a lysine residue. So when you put the aldehyde and amine together, they form what's called a shift base, and this is a covalent reaction. And that's how the 11-cis retinol is bound here to the opsin molecule. And then together, these two are known as rhodopsin, right? Okay. All right. So what else did I want to say about that? Yeah, so what happens when light comes in, we call this photoisomerization of the 11-cis retinol is converted to 11-trans retinol, right? And then the cell bodies, we already talked about inner segments, just what we thought about, what we think about when we think of cells, nucleus, mitochondria, and protein synthesis occurs here. All the things we would think of with normal cell body. But these cells are extremely active, right? They have to continually be making proteins because those discs are shed every few weeks. So they need to make more of the rhodopsin, more of the proteins that are in the membrane of these discs. So very active. Then these cells, the rods, they form synapses with the intermediate neurons and the sensory neurons in the retina. And here's something else I thought that was interesting. Glutamate's a neurotransmitter in this case, but instead of exciting those cells, it actually inhibits the cells. So we're going, instead of depolarizing cells like we would normally think of for the signal to be transduced, the cells are already quite positive in nature because they have these channels on them that are sodium and calcium channels, and they're active when there's a lot of cyclic GMP. So in the dark, when those rod cells, those rhodopsins not activated, there's a lot more cyclic GMP. Cyclic GMP binds to these channels and allows them to stay open. So the inside of the cell is much more positive than what in other parts of the body. I think the resting memory potential they gave in the book was like -30 or something. And what are we used to for neurons? 20?
Testing me. I want to say like -70.
Yes,
Yeah.
-70. Yeah, we're used to like -70. And even at our skeletal muscles, I believe it's even less, -90, something like that. Yeah. So -30 is quite already depolarized in a way than what we're used to. So when the light activates the rhodopsin and 11-cis retinol is converted to 11-trans retinol, cyclic CMP levels go down. So again, remember we talked about how that's different than what we're used to. We're used to the second messenger levels going up. And then because cyclic GMP is necessary to keep those channels open and that levels goes down, those channels close and it actually causes a hyperpolarization of the cell. And then the glutamate is, so like I said, we usually think about glutamate as being inhibitory, but there are two types of glutamate receptors that are excitatory. And they are metabotropic receptors, so they're G protein coupled receptors as well. And in this case, the glutamate is acting as an inhibitory neurotransmitter rather than an excitatory neurotransmitter. So, let's see. Oh yeah, I told you that the extremities of the outer segment of the rods protrude into the layer of cells that form the pigment epithelial. So here we see that, right? How they protrude into those pigment epithelial there. And the function for these is, like I said, they contain the enzymes that regenerate the 11-cis retinol. So regenerate 11-cis retinol. And they, yeah, okay, yeah. They also, what's interesting about these pigment epithelial, do you know why they're called pigment epithelial? Anyone? Well, it's because they already have a color because they contain melanin. What other cells do we know contain melanin?
Skin.
Skin, right? Yep. What else? Yeah, but think of our brain. What other cells contain? Hmm? No. None.
Nigra.
Niagara strata.
Substantia nigra.
I
Yeah.
don't know that.
You didn't know that? Come on! Aren't you in Dr. Leek's lab?
Yeah.
So the substantia nigra, right? If you were to look at them, they have melanin. They're dark in
I
color.
don't know the substantia nigra contains melanin. I don't know.
You don't know this? Oh, you don't know. But you know what the substantia nigra.
Obviously.
Okay, all right, yeah. So let me just see if I can show you a picture quickly. Matt should know this, right? Yes, right? Okay, let's see.
I didn't know that it's melanin that's causing
Ah,
it to be dark. I
okay.
just know that it's dark because
Yeah, so it's
the
called
term
substantia nigra is
Yeah.
dark, means
Mm.
dark
Yeah.
substance. I'm trying to find. Haha. So, oh, well, that's kind of it. But I'm trying to find a real one. Oh, here. See those dark containing cells? Those are the dopamine neurons, right?
Yeah.
This is showing that this is Parkinson's disease, right? So you lose a lot of the pigment because those cells are dying. But they're naturally pigmented like that. They actually look dark in the brain, and that's because of melanin in the substantia nigra cells. Yep. So here's a dot, a picture. Like, see how they look dark there? Yeah. Yep.
What's the role of melanin
Yeah,
in
it's
the
made
substantia nigra? I mean, in the skin,
Yeah.
it's for protection from sunlight, but I was wondering.
So I don't know. I don't know the latest research on it, but when we were, I mean, I don't know if it's maybe to protect them from oxidative stress, but they are more susceptible to oxidative stress than the VTA, which doesn't have the color, the dopaminergic neurons. So I don't know. Do you know the latest research on what melanin's doing in the substantia nigra? Anyone? I don't know. There's a lot of interest in that though, because the substantia nigra cells are more susceptible in Parkinson's disease than the VTA. So it was thought maybe for a while it's because of the melanin, but I don't know. I don't know the latest research on that. I don't know. Yeah. So, oops. Okay, so they contain melanin. So it makes sense though in these cells, right? Because what that means then is some of the light's not going to make it to those rod cells and make it to the rhodopsin. So the melanin is there to absorb the light that gets past the discs and past the rods, and so it prevents it then, instead of reflecting off the cell and being sent back to the rod cells, which could blur. Can you imagine that would be like a blurry vision type thing? You wouldn't be great vision. So it absorbs that light so that does not reflect back onto the photoreceptors. Interestingly, in people who have are albino or have albinism, they do not synthesize melanin, right? So they're not going to have the melanin in their pigment epithelial. So that's why they have vision problems. A lot of people with albinism, especially in bright light, because the bright light is going to scatter more of that light back onto the photoreceptors, and so they have their vision's affected by light. Yeah. Okay. Oh, this is another showing the shift base with the lysine here. in opsin these photos. So then we already told you the G protein is called G transducin. It's coupled to rhodopsin, and it activates this cyclic GMP phosphodiesterase. The cyclic GMP phosphodiesterase consists of an alpha, beta, and 2 gamma subunits. I think it's best just to look at this picture here. So what happens here? So here, the circles, the blue circles are the G protein, right? The catalytic subunits here in this enzyme are alpha and beta. So these are the catalytic subunits. And the 2 gamma subunits are inhibitory. So when there's no G protein bound to it, activate G protein bound to it, the enzymatic activity of the cyclic GMP phosphodiesterase is inhibited, right? But then you get the activation of the rhodopsin. Here, this is showing the activated rhodopsin. And, oh, by the way, yeah. So it shows you up here in this photo up top that the rhodopsin, the activated form is this metarhodopsin 2 here, right? Okay. But before it gets to that final activated form, it actually goes through a couple of different transformations, and it actually takes up to seconds to get to the activated form, or milliseconds to get to the activated form. But once that form of rhodopsin is activated, the G protein is activated, and then it binds to this cyclic GMP phosphodiesterase, and it binds to these 2 gamma subunits that are inhibitory, and in doing so, it causes a conformational shift that you can see in the figure on the right there, and allows the catalytic sites, the alpha and beta sites of that enzyme to be exposed, right? Yes. Removes the inhibitory subunits, so that then the cyclic GMP can be converted to GMP. So instead of increasing the amount of cyclic GMP, we're getting a decrease in the amount of cyclic GMP.
It's that, I mean, it takes 60 seconds to convert all this. Is it like, maybe this is the reason in the dark we need some time to adjust our eyes?
Yes, that's right.
I'm not sure.
It's not an instant thing. Yeah, I think that's why. Because, 60 seconds to get to the opsin plus all-trans retinol. Yeah. It's faster to get the rhodopsin activated, but it's still not as fast as you would think. It's not like nanoseconds. It's more like, microseconds.
Yeah, you can count the time in head, like, okay.
Right, yes, exactly.
It's not 60 seconds.
Not 60 seconds, but it takes some time. Like if you suddenly the light goes off, you can't see anything. But with time,
60 seconds is too much.
60 seconds is too much. I know.
Yeah, it's not. Yeah, it's 60 seconds to get to all of it to be converted to the opsin.
Gradually, you can see like it happens gradually.
Yes.
The wavelength that it's absorbed.
Yeah, it starts 10 to the minus 12th, and by the time we get to activated rhodopsin, it's 10 to the minus 3rd. So what's that? That's like milliseconds, right? So something you can comprehend versus 10 to the minus 12 seconds. You can't even comprehend that, right? So it is. So it does. That's why it takes some time when the lights go off. Mm-hmm. That at first you can't really see, but if you stay there for 30 seconds, I don't know, maybe up to a minute as you get older, then you can finally kind of see your things in the dark. It's really freaky. You don't have to do this anymore, but what was always really freaky to me was we used to have to, for slides, since we didn't have PowerPoint, that's how old I am, we'd have to take pictures, like photographs, of our data, and then we'd have to develop. So at one part you had to be in a completely, like you could have the red light on sometimes, but at some point you had to be in complete darkness, like no light, and it freaked me out so much because I had no orientation of anything. You know what I mean? You just have if you ever get a chance to be in the complete dark, it's so freaky. You know, we don't often get a chance to be in the absolute complete dark, but in those dark rooms and you turn out the light, especially if you stand there for a minute or two, because at first you're like, okay, I know this is over there. You know, if you close your eyes, you know this is over there, this is over there. But then when you stand there for a few minutes, you're like, is this really over here? Because I can't can't see it. You know what I mean? It's very disorienting. It's supposedly good for you. You ever hear of those chambers, those sensory chambers that they put you in, where you float? Basically, you float in salt water or something, and there's like, you can't hear, you can't see, you can breathe obviously, and you can't smell anything. So it's all sensory deprivation. It's supposed to be like really good for you, like soothing, to like turn that all off for a while. There's places in Pittsburgh where you can go do it. Actually, you can go float in these tanks, and
I think I would panic.
Right. That's what I'm saying. I think I would panic.
I can't.
Has anyone ever done it? Jared, do you know anyone who's done it? Yeah. But there are places here you can do it. It's
I doubt it can give any calm to anyone.
I don't know. It depends.
Some people maybe.
It depends. I think my husband would benefit from it because he gets overstimulated very easily. He's a he's introverted type person, and so social situations. He's better. He's 53 now, so he's learned how to deal with it. But social situations used to freak him out. I'm complete opposite, by the way. Complete opposite. I walk into a party and I'm like, hey, I know everyone but in the first 10 minutes, and he's like, oh my God, look at that. I'm gonna go just hide into the corner. So I think he would benefit from some of that. I think the sensory information of this world gets to him a lot. So
because I remember we went to a place, it's a cave, very historical cave, and everyone went in there. Like, I was claustrophobic the minute I went in. And halfway through, I had to tell the guide, like, let me out. I don't
so
need
you
this
have
experience.
it's not just dark, it's claustrophobia.
Everything. Like, if I want to relax, I need open space. It's okay. I cannot think myself putting in a tank. It's just gone.
Interesting. Yeah. Phobias are interesting.
Yeah.
Even thinking of that makes me more like.
Are you claustrophobic too?
I don't know, but I would not survive.
No, right. What do you think? Do you think you would?
I don't know. Like, I have to experience it.
Yeah.
Love to experience.
And
What about
How are you, Young? You want to do
small
it?
place?
I've been the cave.
Yeah.
I did fear
Laurel Caves,
Yeah.
Laurel Canyons, and
they kind of put me in the
I felt like
and then the sound is
amazing.
very bad inside.
yeah, you felt good.
Yeah.
You liked it.
Yeah.
So Young would like it, I think. He says he likes the smaller spaces, right?
I mean,
That's good.
that was adventurous.
Adventurous.
Like
So a lot of adrenaline.
Yeah.
Yeah, I like the caves and stuff.
Even you see videos of the caves, people going in small caves, very small opening, going in with all darkness. I cannot even see those videos at
Yes.
all.
Really?
Same.
Oh my gosh, that's funny.
I had an experience that one in the Philippines because we had this class that we were required to like go on a field trip, and then the kids like have an option to go through the tunnel, and then
Did you do it?
I did. Just because we wanted to experience it. So like, everyone went through that, but I don't see any advantage of going.
What about you, Matt?
I mean, like the sensory deprivation tank. I think I would try.
Yeah.
I've seen them around. I think there might be one in like in Bloomfield, like right by my
Yeah, I'm
house.
sure they're close by. Yeah, there's other all over.
I think it'll be like very weird. Cause like after a while, like do you think like you like just start hearing like your heartbeat?
Right.
Stuff,
Yeah.
is it, like,
I would
you get
imagine
adjusted to
because
it?
they can't. I don't think they can get out all sound unless I don't know. I mean, because your internal.
Right.
Yeah, I think that's part of it. Is you're supposed to get in tune with your like your heartbeat, your breathing. You know, that's what you're supposed to be hearing rather than all the external noise. Yeah, it's crazy. We in Pacific Northwest, my husband and I went in a cave, and we didn't realize this, but the guide's like, you can keep going. I'm not going to go with you. You can go like another, I don't know, he said like another 100, 200 yards. And we're like, okay. But eventually it got to the point where we were literally like on our bellies like worms, and I'm like, yeah, we gotta go back. We don't gotta be here. This is what are we even doing? I mean, it's not even, I don't even see anything. Like, it was crazy. So
That I would absolutely.
yeah, that was
Yeah.
that
that's
was
amazing.
Never.
that was insane. Let's see, what else do I want to talk about with this? So, contain melanin, absorb light, the transducin. Oh, we're talking about this. I think that's it. I mean, the book also talks about how calcium regulates it, but I didn't include that on purpose because we're going to do calcium regulation, calcium signaling in the next couple weeks, which I want to talk to you about. So, first, before we do that, any questions about this visual stuff? I just think it was an interesting case about how it's very different from the other G proteins that we studied. Who knows? Maybe a G protein will pop up somewhere else in the body that acts in similar ways. We never know what we'll find, right? So I just thought it was interesting. So, any questions about this? Okay, so what I want to do for the rest of the class, let me just go to your Canvas site here. Okay. All right, so we did chapter 4, 5, 6. We still have 7, 8, 9, and 12 that I would like to at least cover before we adjourn for the class. And I know that Dr. Johnson did his part. Did he do pharmacogenetics or pharmacogenomics with you? No. Okay, he was supposed to. Well, anyway, we'll forget. He didn't finish his notes, but I can't worry about that. So what I wanted to do is I wanted to go through still calcium, because I think calcium is a really important signaling molecule and effector. I wanted to talk a little bit more about different types of PKA, PKC, how they're regulated, and then of course the receptor, the protein tyrosine kinase receptors, right? Okay, and the signaling. So you all are going to be the teachers, okay? And this is going to be in lieu of your final exam, okay? But it's instead of so for most part, like your exam, I'm going to grade. When you did your presentations, that was most part just do it, and you get the full points. It's just for this is actually going to be graded, but it's going to be like seminar class kind of. I'm going to let all of you kind of grade each other, okay? So I'll make a rubric for that before we start. So we're going to skip class on Thursday so you can get prepared for this. So there's 6 of you, and the longest chapter is chapter 12. Okay, so I think that 2 of you can do the calcium stuff together, the intracellular calcium and calcium effectors, right? 2 of you could do the, or 1 of you could do the PK and PKC. I don't think that that's a very long chapter. This is how I had it. Wait, there's 6 of you. 2, 4. Oh no, 2 of you could do PK and PKC, and 2 can do the receptor tyrosine kinase, chapter 12. Okay, I don't care how you decide who's going to work together. It's fine with me. Obviously, the intercellular calcium would be up first. So if you want to go first and get it over with, that would be.
Can I go?
You want to go and get it over with?
Yeah.
Okay, so
Yeah,
Young.
I would like to go because I'll be in India.
Oh, okay.
All right.
Is that okay with everyone if Young and
I'm
Vivek?
going next. I won't be here next week.
Oh yeah, but he's not.
You won't be here
Because
next
of
week?
DC, SFN.
Oh, that's fine. So that's good. So you won't. Next week would be calcium. So these 2 could go, right?
Yeah.
Is that right?
Yep.
Yeah.
Is that fine? And then PKA and PKC.
I can do PK, PKC.
Do someone do it with her, Melanie, and then you 2 work together.
I think I would go in the last.
The last.
Yeah.
So that would be receptor tyrosine kinase. Is that okay with you too? Okay. So what I want you to do is the chapters are here for you to read. Like I did with the visual transduction, you don't have to say every single part of the chapter, but make a handout, kind of like I did here, that you're going to walk us through and give us the pertinent information. and then also find one primary research article that relates to your chapter. Okay, and then talk about how that relates to the chapter. Maybe how you could use it in research, not your research, but any research. Things how else, how you would summarize a research article. What's the importance here? Does it relate to, how does it relate to research and drug discovery or whatever, okay? And then I'm going to have a rubric for everyone here in the class listening to your presentation, and we're all going to, of course I have the final say on the grade, but still we're all going to give constructive criticism to each other, okay?
Yep.
And then that'll be in lieu of a final exam. This will be like your final exam.
There's no other exam.
No other exam other than this.
Dr. Johnson is not taking any exam.
I don't think so. I'll talk to him.
Okay.
I think I think he's kind of done. Did he say he was going to do a final? No. I'm going to tell him this is what I've decided, and he'll be like, okay. I sent him a really long email before, like, this is what I'm thinking of for the course. After we met a week or two, he wrote back, that sounds fine. So I don't think he'll care. So I'll double check with him, but I'm pretty sure he won't have anything extra to give you.
So Dr. Khan, how
Yes.
are we distributing like the topics among ourselves? Like in intercellular calcium, does like we do intercellular calcium, and then because there are like 4 chapters.
Well, the calcium effectors, I thought maybe you could put that together with your
yeah.
intracellular calcium, okay?
Yep.
Yeah?
Yep. So I can go for the effector side. If you wanna go for the first one, intracellular calcium, or the other way around is fine.
No, it's fine.
Okay, then I'll go.
You take the
first.
effectors,
Okay.
I take the intercellular
Okay.
calcium.
Okay?
Yep.
So we won't have class Thursday, so you can use that time if you want to figure out how you're going to do it, what you're going to do, okay? So teach the class and one primary research paper that matches your topic, that relates it all to maybe to a drug mechanism, yes. To bring it all back to drug mechanisms, okay?
Yep.
14, maybe.
So the next class will be
Next.
next Tuesday,
14.
right?
Yes. 14.
Yes, 14. Okay?
Yes.
So after that, Melanie and I will present on 16th.
Yes, let me, yeah, so the idea is, since we want to get this all done as soon as possible, let's look at the,
Then the Thanksgiving break.
right, so next week will be calcium, and if we get through on Thursday, you would do the PK and PKC, and then it's Thanksgiving, and then the week after Thanksgiving,
28.
Tuesday, December 5th, we'll do the tyrosine kinases. It might go to Thursday, December 7th, but let's try to get it done by Tuesday, December 5th.
Yep,
Then
yep,
we're
yep,
done.
yep, yep.
Oh wait, Thursday we won't have class, so it's just Tuesday, December.
Okay.
Oh wait, no, we have that week of November. I'm sorry,
So we also
We-
have like 27 or 28.
28th.
Tuesday.
That's right. I'm sorry. We have the 28th and 30th.
Mm.
Well, we'll see how these first 2 weeks. So it'll be. We can even finish before December. We can finish
Yep,
end of
yep.
November.
Yep.
Just be